Go. And welcome to another wonderful day of Bio 202 in the neighborhood. I'm Mrs. Gillespie, your in the shade teacher, but you won't need to look at me anymore. And, oh, I know. No, that didn't do it either. Oh, well. Okay, so today we are starting the urinary system. I hope that you have taken your test, or you're going to take your chapter 4 test, and that you do well in that, or not chapter 4. Session 4, whatever, test number 4. So we'll just go right into urinary system. I'm not going to send you announcements that class is starting anymore, because you're obviously here, and so if you're not here, you're probably not going to be here. So anyway, so, and remember, oh yeah, everything's in modules. So don't be, I took away your ability to look at quizzes. I took away your ability to look at assignments because you were getting confused at where do I submit things. Just go to the module will guide you, do everything in the module, and you'll be perfectly fine. So all right, so we're starting module 5 in the urinary system, which is that's all it is. It's just urinary system and fluids and acid base, but it's all together. So okay, so let's get started. Oh, I forgot to mute you people when you come in, so you don't have to worry about it. Okay, all right, and away we go. All right, so we've done some of this stuff already when we did lab, like the anatomy stuff, so I'm going to skip through maybe a lot of it and in the beginning. So here's where the kidneys are. We see that they're in the lumbar region, so just below your rib cage, or your rib cage kind of covers them. The last two, the floating ribs, kind of cover over your kidneys. Remember that you have adrenal glands on top of them. You have renal vessels going to and from them. You have the ureters that are attached to the bladder and or insert into the urinary bladder. I also mentioned embryonically they form down here right with your gonads. The gonads and the kidneys form from the same embryonic tissue, and then your kidneys move up into your abdominal cavity. Yes, one is higher than the other, because if you look at here's on the right, this is where your liver is. So remember, because they ascend, the liver was already there, and so the right one couldn't go as high as the left one because the liver was in the way. Okay, so they are retroperitoneal, which means they are behind the peritoneal cavity because they formed separately and independently, and it also makes it so that they're protected from anything that might happen in the peritoneal cavity, any kind of infection. If my appendix ruptured, for example, that infection could not get into my kidneys. And they're also surrounded by fat. So we have this perirenal fat that's outside of here that helps cushion it. And then you can see down here in this picture how they are covered slightly by the 11th and 12th ribs. Okay, let's see. So we looked at this. Whoops. Let me just go back here. So remember that we have the cortex on the outside, we have the medulla on the inside, we have the renal columns where the blood vessels that come in and out from the hilum are going to cut through the medulla and leave these renal columns. So you can actually see in this real picture of a kidney that there are blood vessels in these renal columns. And then what that's going to do is section off the medulla into triangles that we call the renal pyramids. And at the end of the renal pyramids are the papillae, where you can see, oh, this urine that's been stained yellow is actually, well, I think it's latex, but anyway, is coming out of the papillae, the very tips of the pyramids, and collecting in the calyces. So we would have calyces, 5 or 6 minor calyces, and then 2 or 3 major calyces that are all going to come together into the renal pelvis, and then that's going to become the ureter. So this picture just shows how the blood vessels are going to pass through the renal columns on their way out to the cortex, because all of filtration and reabsorption is going to happen within the cortex. As far as blood vessels are concerned, okay, so we've got the glomerulus, the little ball of capillaries, so it's a ball of yarn, that's what it means. From the afferent arteriole, the capsule is blind because it's like a fist in a balloon again. So that's a really good image. So let's say that your fist is the glomerulus and the balloon is the glomerular capsule, so we just push into it, so there'll be a part of the balloon that's going to touch the glomerular capillaries, and then there's going to be a cavity, and then there's going to be the outer wall of the balloon, and then the opening where you inflated the balloon would be connected to the proximal convoluted tubule. Let's see, so it's going to entirely surround the glomerulus, we know that. Proximal convoluted tubule, simple cuboidal epithelium, mitochondria, and microvilli. Loop of Henle has different characteristics depending on if we're in the thin descending limb, simple squamous epithelium. Thick ascending will be simple cuboidal with some microvilli again. And then distal convoluted tubule, cuboidal cells because they came from the thick ascending limb, but not many microvilli.
So the Bowman's capsule and the
glomerular capsule are the same thing.
Same difference, okay.
Yep. Let's see, so here you can see the simple cuboidal epithelium of the proximal convoluted tubule, the distal convoluted tubule. Here's the descending limb of the loop of Henle. The ascending limb would look just like the distal convoluted tubule, and then the collecting duct, you can see very few mitochondria because we're not really going to do any active transport with the medullary collecting duct cells. Yeah, some cells with abundant if they're in the cortical part, but the medullary part aren't going to have any. Peritubular capillary bed came from the efferent arteriole, surrounds the nephron for absorption and secretion. Afferent arteriole, okay, so we got all those. We know all those things. Really like this picture because it shows just how convoluted the nephron really is. What's interesting is that this is primarily one nephron that has been so coiled up that when we did the section on it, that we cut through the same nephron. How many times did we cut through the nephron? So you can tell proximal convoluted tubules are ones that still have material to be absorbed. So you can see all this filtrate that's in these tubules here. And then the ones that are more clear are the distal convoluted tubules. And the cells are a little more square with central nuclei. You can see them a little more distinctly in the proximal or in the distal convoluted tubule, and not so much in the proximal. Glomeruli are cool. Here's a glomerulus here. You can actually see the red blood cells in the glomerulus, all the capillaries, and here's another glomerulus over here, and there's just a little hint of one over there. An artery that's afferent arteriole, probably carrying blood to these artery, or to these whatever they are, glomeruli. Oh, and here's another thing. Let me draw this because this is kind of cool too. Let me get in a color you'll actually see. Let's do green. Here you can see the Bowman's capsule really good. Don't call it that, the glomerular capsule. All right, so here it is, and then it's going to be going into, so this is a proximal convoluted tubule over here, and then here, and then, okay, so then it's going to come around, and then this would be the visceral border, but remember with the visceral border that it's all going to be covering the capillaries, and then this region right here is where the afferent and efferent arterioles are. And then this probably, I would venture a guess that this right here is the distal convoluted tubule with the macula densa right here. So remember when we saw in lab that the macula densa is going to be right here at the beginning because that's the one that checks what the filtrate, the composition of the filtrate is, so that we can fine tune what our filtration rate is. So that's really good. Yeah, I'm pretty sure I used to be able to really well identify that, but that was last year when I was doing histology. Okay, so let's get rid of those. And all right, so we got two kinds of nephrons. We have juxtamedullary nephrons and cortical nephrons. So the cortical nephrons are the ones that are primarily within the cortex. So this one right here, okay, that's going to be a cortical nephron because you can, oh, see right here, cortical nephron, because most of it is found within the cortex. The juxtamedullary means to place right next to, like juxtaposition. Well, juxta means next to. So this is next to the medulla. And so then we have this long loop of Henle that's going to extend down into the medulla. And when we get into the formation of urine, and we'll talk more about the difference between these two. It's easy to see the border between the cortex and the medulla, because this would all be cortex up this way, and then medulla down this way, because we have these two arcuate vessels, arcuate artery and vein, and they just run horizontally across between at the border of the cortex and the medulla. So it's easily identified there. All right. Oops. Oh, let's go back to the notes. Okay. Oh, here's one thing. So the cortical nephrons found almost entirely within the cortex with the paratubular network, capillary network. Juxtamedullary nephrons found in the cortex and medulla. Loop of Henle has a vasa recta instead of a paratubular capillary network. So we have the paratubular capillaries up around the proximal and distal convoluted tubules. We have paratubulars around the loop of Henle here, but look at in the juxtamedullary nephron that we find a vasa recta. Okay, and I talked about that last week in Tuesday's addendum to our lab. So make sure you know the vasa recta is only with the juxtamedullary nephrons, and it's only with the loop of Henle. Otherwise, it's paratubular capillary network. Here's just a nice picture of what the glomerulus looks like in afferent and efferent arterioles. So here's the afferent going in the glomerulus. Here's the efferent coming out in each of those. Okay, so we're going to have fairly high blood pressure, okay, vascular resistance. As we come into the kidney and the renal artery, mostly so we don't pop the glomeruli, so we're not like just flooding the nephrons with blood. And so we need a means by which we can regulate that. And so we started talking about it in lab, but now we're going to get into a lot more regulation of, specific processes that occur that are going to regulate our blood pressure. But I just realized before we go into there, I forgot to do what are the five functions and 3 processes of the kidney, so of the urinary system. That's how I always start this, but you know what, I'm not in front of my board, so I'm going to stop sharing that and go to my whiteboard. And let's see, let's go to text. Okay, so yes, functions of the urinary system. Okay. All right, so this is participatory. So somebody just either chat in or unmute yourself or something and give me a function of the urinary system.
To make urine.
Okay, yay. Okay, and what is urine? It's a excretory product that removes. Okay, what are the 2 main things we want to, well, 3. We'll do 3 things that we want to remove from the body into our urine.
Acids?
Okay, yes, okay.
Nitrogen.
To remove, I'm going to put H+. Okay, and?
Nitrogen.
Okay, so our nitrogenous wastes, so urea and uric acid. And one more thing. Oh,
creatinine?
That's going to be in nitrogenous waste.
Oh, yeah.
Bryce said urine in the text thing.
Oh, Bryce said water, and Bryce is right. Oops, as I'm texting back in the chat. Yep. So those are the 3 main things that we want to get rid of in our urine. So hydrogen ions, so we want to control our acid-base balance. We want to get rid of our nitrogenous waste. And so creatine, or creatinine, I mean, that's one of your blood tests. So if you have like a bun, so you have blood, urea, nitrogen, and then you have a creatinine levels that we're testing in our blood to give us an idea of what kidney function is. So if you've ever had a blood test done and you've looked on there and there's been the BUN, so capital B, capital U, capital N, and then creatinine, that's what they're for. We're looking to see how well your kidneys are getting rid of nitrogenous waste. And then water, obviously, okay? So we drink lots of water and we need to get rid of it or we'll be edematous and we'll all bloat and that wouldn't be good. Okay, what is a second function? Put the little chat up so I can still see it.
Blood pressure regulation.
Yay, because that is exactly right. Okay, so blood pressure regulation, and I'm going to put in parentheses long term because we're going to discuss exactly long-term blood pressure regulation today. Not today, but in this unit. But that's
blood acidity.
okay. So
Regulation.
yeah. So acid-base balance, homeostasis. Okay. We got two more. How about this one? Okay, fluid and electrolyte, whoops, homeostasis. And one more.
Red blood cell production.
Yay. So you printed out your notes. I'm so proud of you.
Yep.
Stimulus, let's put it that way. Stimulus of red blood cell production through what?
Secretion of erythropoietin.
Yeah. Whoops.
I really did print out my notes.
Okay, perfect. All right, so those are the 5 functions. And so now we want to look at how these things are carried out. So what processes happen, and you guys already know this because we already talked about this. So what are the 3 processes of the urinary system? Yes, filtration, good, Bryce? Oh, please. Did you die? Am I still here?
Secretion and absorption.
Reabsorption. I just was worried there for a minute. I don't know that you're not gone or something. You know, you could have been the 144,000 and the rapture happened and I'm still sitting here going, hello?
No, I accidentally clicked on the text box and was pushing the space bar down and I was just going as I was talking to myself.
Yeah. Okay, so there we go. Filtration, reabsorption, and secretion. So through those 3 processes, we're going to be able to do these 4 over here, except for the 5th one. So the secretion of erythropoietin is independent of these 3 processes. So it's just going to be, well, I guess it's still secretion, but that's not tubular secretion. So a little bit different. All right. Anybody have any questions on this? So please remember this for ever. Don't ever forget it. Okay, so let's go back to now our juxtaglomerular apparatus and see what that's all about. Sorry, I'm still trying to finish my breakfast. All right, so let's look at a picture first, and then we'll go back to the definitions. Okay, so here is our juxtaglomerular apparatus, all of this little area right here. Not the mesangial cells. Well, yeah, I don't really get into talking about those, but they do have a function. But just right here, we're just going to go with these guys right here. So we have an afferent arteriole. Surrounding the afferent arteriole, we have juxtaglomerular cells, also known as granular cells. So in the recording thing of the interactive physiology that I showed you last week, granular cells, juxtaglomerular cells, they're the exact same thing. And then we have the efferent arteriole. We have some around there, but primarily we want the JG cells around the afferent arteriole. And then we have the distal convoluted tubule. Well, thick ascending as it's turning into distal convoluted tubule. And then right here, super important that you remember that these cells right here, these right there, the macula densa, are chemoreceptors. Okay, because they're going to detect the sodium chloride concentration within the filtrate to see if any additional modification of pressure needs to be done in order to make sure that GFR is where it's supposed to be. So one very important thing about the JG cells is that they secrete renin. So renin is a hormone, yeah, it's a hormone, but it's more paracrine than it's actually endocrine, like going through your whole entire body. We only need to find, once we secrete renin, we just need to find the angiotensin or angiotensinogen and then get that whole system going. But anyway, it's a hormone. The JG cells also act as mechanoreceptors that directly sense the blood pressure in the afferent arteriole. Whoops, I went the wrong way. So as the afferent arteriole stretches, then it also lets, or doesn't stretch, as the case may be, lets the juxtaglomerular cells know, oh, pressure's high, pressure's low. Okay, then the next, so here's another good view. Oh, this is a really good view. I know, this was from my histology class. No, my prints, well, anyway, one of my classes I took last year. And so I like this a lot. So I want to show you how this works. I want it thicker right here. So right here is just going to be regular simple squamous epithelial cells. Okay, just right to here. On the outer border of the parietal wall of the glomerular capsule. And then we change into the simple cuboidal right here, down here. So you can see how they've turned into simple cuboidal. All right, so now though, what's gonna happen is that we get these cells called podocytes. And so these little triangles, these little gray triangles are actually cell processes. Okay, so here's the cell body of a podocyte. And so it's gonna have these little fingery things that then wrap themselves around the capillary. Oops. Okay, so they wrap themselves around the capillaries and then interdigitate so that, oh no, that'll work. So that now through the filtration slit, I'm going to show you where the filtrate's going to go. So remember this, oh, hang on. Gonna get a color. Okay, so this down here, this is my fenestrated capillary wall. So here is my capillary. So these gray dashes, now they're red. All right. Okay, so now here's where the water's going to go through. The filtrate's going to go through. So it's going to be right here. So this is a cross section of a capillary and in the glomerulus. And so then it's going to go through here, go through the basement membrane, and then it's going to find a filtration slit and it's going to go out there. Going to go in here, find a filtration slit, go find a fenestration, find a filtration slit. Okay, so you guys can see, so this is a really good representation of the glomerular capsule and its relationship to the glomeruli. So all of these PO cells, all these black or these gray blobs, so these are the cells, and then they just have all these little arms that go and wrap around the capillaries. Okay, so that's all I wanted to do on that one. All right, so this shows, what are we looking at? Okay, so I put this one into, that was from my histology class. Here is the body of the podocyte, cell body. right here. And then see how they have these arms, and then the arms just branch into these little finger things that are going to wrap around. So all of these black right here, these are all the filtration slits. So that's where the actual filtrate's going to come into. So in this picture, we're swimming in filtrate. So we would be immersed in the filtrate because it would be coming out through these holes right here. But I really like that scanning electron micrograph. Okay, so then also in the juxtaglomerular apparatus, we have the macula densa in the distal convoluted tubule. Some people say the thick ascending limb, but it's right at the top, just as thick ascending is turning into proximal convoluted or distal convoluted tubule. So there it is, chemoreceptors that respond to the changes in solute concentration of the filtrate. We did this really good now. We've got this down. So between the blood and glomerular capsule, it's porous to allow free passage of water and solutes smaller than plasma proteins. Similar to the respiratory membrane in thickness and function. So let's go back to this picture right here. So the filtration membrane would consist of the simple squamous epithelium and its fenestrations in the glomerular capillary wall. And then this gray, this darker gray right here is all basement membrane. And then these little gray, the podocyte, fingery things, and filtration slits in there. So again, you can see it's very, very thin, just like we had a very, very thin respiratory membrane. All right. Okay, so there's just a drawing of it, representation of it. So I think we're good with that. And another one. Oh, this is a nice picture. This shows the pathway of the filtrate. So here it is in the plasma. Here are the fenestrations in the capillary wall. Here's the basement membrane, and here are the podocytes, the little foot processes. So these little fingery things are the feathery looking stuff right there, and then the filtration slits. All right, so we're going to take a look at our processes now, in especially filtration right now, and taking a look at how do we form the filtrate. So every minute we have about 1 liter of blood passing through the collective glomeruli. If we have a heart rate of 72 beats per minute. We already know that we have a glomerular filtration rate. So out of the total glomeruli, 125 milliliters is forced out into the tubules. So we're going into the tubules. The filtrate in the tubules is everything in the plasma except the proteins in the cells, because they will not fit through the pores or the filtration slits. We want to keep them behind. So that's filtrate. So make sure you know the difference between filtrate and plasma, and not plasma, because they're going to be the same thing. Filtrate and urine, because they're two entirely different things. They're formed, filtrate gives rise to urine, but they're not the same material. So urine is just everything that we want to get rid of. Our metabolic waste, unneeded substances, extra water, all of that, okay? All right, and so if we're filtering 125 milliliters per minute, 180 liters of plasma will be processed every day. But look at that, only 1%, okay, that's just under 1% of, so 1.5 liters leaves the body as urine. So remember, we filter, of the 100% of filtrate that we produced, 99% of it is reabsorbed, leaving 1% eliminated every day. And so the processes that allow this to happen are glomerular filtration, tubular reabsorption, and secretion. All right. Oops. Okay. Here we go. So here just shows where those processes are happening. Remember how we've already said filtration is capillaries, or is blood to tubule, reabsorption is tubule to blood, and secretion is blood to tubule. So these two don't, this occurs, so this tubular reabsorption is primarily in the proximal convoluted tubule. Let's go ahead and write that. Okay, so PCT is where we do tubular reabsorption, but this can happen just about anywhere. Proximal convoluted tubule, loop of Henle, distal convoluted tubule. It just depends on where we need it to happen, where the mechanism for it to occur is found. All right, so filtration then is passive because we're not forcing it. Well, we're not pumping it. So let's put it that way. We're forcing it through hydrostatic pressure, but we're not pumping it. Okay. Non-selective. That means everything smaller than the whole is going to get through to be found in the filtrate. Okay. And hydrostatic pressure is the driving force. By keeping albumin in the plasma of the capillaries, that creates colloid osmotic pressure. And so this way, as filtrate is forming, water is being drawn back into the plasma, and so our the blood in the glomerulus doesn't become too viscous so that it can't flow anymore and can't return back to circulation. Remember we said last week too, the presence of presence of protein and or blood cells in the urine usually indicates some problem with the filtration membrane. All right, so we had some terms last week. So we had GFR of 125 milliliters per minute, but what caused a GFR of 125 milliliters per minute? An NFP, or a net filtration pressure of 17 millimeters of mercury. What creates a net filtration pressure of 17 millimeters of mercury? Hydrostatic pressure of the glomerulus. Okay, so hydrostatic pressure of the glomerulus pushes water and solutes across the membrane into the capsule. Osmotic pressure of the glomerulus pulls the water back into the capsule, and hydrostatic pressure of the capsule pushes filtrate back into the glomerulus from the capsule. So here's what happens. So in the purple arrows, we can see that's the hydrostatic pressure out. Because the filtrate doesn't all immediately drain away, there is some pushback from the filtrate inside the capsule. So that's capsular hydrostatic pressure. And then this orange colored arrow is what's drawing the water back into the capsular into the glomerulus due to the presence of albumin. Okay, or the presence of albumin. So here's just some numbers. This would give you a net filtration pressure of 10 right here, because you would add 30 and 15 and then subtract it from 55. So 55 minus 45 gives you 10. Okay, so a GFR of 125 milliliters per minute gives you 7.5 liters of fluid filtered in an hour. So that's all of your blood supply, and then half again. So 1.5 times your blood is filtered in an hour, and that's why it works out to be about 36 times in a day that you filter your blood. What allows such good glomerular filtration rate is because we have enormous, and I yawned too in the middle of that. Enormous surface area availability, filtration permeability also. And then here's one thing to remember with net filtration pressure: even just a 15% drop in filtration pressure can stop the filtration altogether. So like we talked about last week, if you're dehydrated and you don't have, so if you went from a GFR of 60 to a GFR of 43, that is going to, that's gonna cause a decrease in filtration to the point where you just can't filter your blood properly. So increase in blood pressure, increase in GFR, dehydration decreases GFR by decreasing blood volume, which also decreases blood pressure. All right, so we're going to come back to this. I don't know why this is right here. We're not going to even start this right now. But remember that in more times than not, because we're in parasympathetic stimulation, we're going to be in a low blood pressure situation. So most of the time our brain is going to be screaming to get some blood up to it because the pressure is not high enough. So we have ample means by which we can raise our blood pressure to get it back to a proper GFR filtration rate. And so we have two ways that we can do it. We can do it intrinsically, which means at the kidney level. So remember we did these last week: myogenic mechanism, tubuloglomerular mechanism. And then we have extrinsic, where we go out and affect the whole body. So we will worry about that later, but eventually, so please know that you need to know every step in this picture at some, well, before the exam. I was going to say at some point, but yeah, that's the point, is that when you take the next step.
Can you go back to that picture really quick?
Yeah.
Miss Jean?
Uh-huh.
Thank you, I got it.
Yeah, and it's in the PowerPoint. I mean, all of this is, I mean, you guys have absolutely everything that you could ever want. Okay, so how we reabsorb is going to depend on how we filter. Okay, so if filtration is too fast, then too much filtrate will be lost to the urine. If it's too slow, then, thank you, Joanna says the picture's on slide 29. So if it's too slow, waste can be reabsorbed, too little blood will be filtered, and we don't want that either. Okay, so, well, I guess we are going to talk about this right now. All right, so we're going to do the intrinsic mechanism. So let me go back to this, the intrinsic mechanism. So that's these two things right here. So within the kidney itself is intrinsic control, where we regulate primarily the diameters of the afferent arterioles, but we also can affect the diameter of the efferent arteriole, okay, through our intrinsic control. So the first one is myogenic. I love this one so very much. So this is the afferent arteriole smooth muscle response. So remember, smooth muscle is going to be like wringing out a towel. Okay, so always vascular smooth muscle, wringing out a towel. So now think about, so your little hands are on your towel and you're getting ready to wring it out, but then that pressure inside of the towel pushes out on your hands and your little fingers are like, no way. And so then you clamp down harder and you twist harder in order to stop that outward pressure that's within the towel. The little weightlifter guy, is he's pushing out those actin and myosin that we have running crisscrossy through the towel, through the vascular smooth muscle, so that it wrings out, you know. That's just going to be just a natural response to smooth muscle. You don't have to send nerves or anything like that. It is just an intrinsic, oh, look, there's that word. It's just an intrinsic characteristic of vascular smooth muscle. Okay, so remember, increasing systemic blood pressure. So I'm going to highlight some things. Okay, so, when we have increasing systemic blood pressure, okay, so then the afferent arterioles will constrict. So why do we want that to happen? Because you'd think, well, that's increasing the blood pressure. No. Well, yes, it is, but that's not what we're after. We are after reducing flow, because remember, increased resistance, reduce flow. If the pressure is too high, I'm going to have too much blood being delivered to the afferent arteriole. So I want to restrict flow and lower glomerular blood pressure. Okay, prevents it from rising to damaging levels. And then conversely, in a decline in systemic blood pressure, then there's not going to be a pressure inside of your smooth muscle anymore. So it's just going to go relax and then it's going to dilate. But then we also have some factors that can cause further dilation if necessary, which we're not going to get into yet. Okay, but right now, if you just stop pushing out on the vascular smooth muscle, then you will dilate the afferent arteriole, which will raise hydrostatic blood pressure by delivering more blood to the glomerulus. Okay, all right, so in both of those ways, we're helping to maintain normal GFR. So what I suggest early on from the outset of learning about this stuff is start writing down little key things and making like little flashcards or something, but don't just look at this big volume of your notes and go, holy crap, where do I start? Okay, so I would just make you a flashcard right now and have myogenic on it and then have 2 myogenic cards. So then one myogenic card on the front, you would put increasing systemic blood pressure, and then on the back causes afferent arterial vasoconstriction. Okay, and then you can also put why do you want that? To reduce blood flow, lower GFR. Then make another card, myogenic mechanism, and then decreasing systemic blood pressure, and then put on the back dilation of afferent arterioles to deliver more blood to the glomerulus. And I would start today, and because there's so much that you have to remember. There's a big volume of stuff that we have to do before the next exam. So getting these key little points and going over them all the time will stick them in your memory, and then you'll be happy. All right. Okay. Now, tubular glomerular feedback. So you could also make 2 more cards for that. What are we going to see in tubular glomerular feedback? All right. So let's look at slow flowing filtrate. Okay. So if we have slow flowing filtrate, then we are going to have a low. So what's our number that we're supposed to have? 125 milliliters per minute. So you can see 90 is less than 125. Okay. So we know that the pressure is not high enough to get us to 125. What are we also going to see? And then, so because it's the macula densa, we've got to look for something. And that's not in my notes. It's down here, but we'll put low NaCl concentration. So I'm not filtering enough. So yeah, I'm only producing 90 milliliters per minute, but what will chemistry, what do our chemoreceptors detect? A low sodium chloride concentration. So what are we going to do if it's slow? We're going to vasodilate again. Okay, vasodilate the afferent arteriole. But there's one other thing that we're going to do in whenever our blood pressure is low, we're going to trigger the renin-angiotensin mechanism. But here's the problem with renin-angiotensin mechanism is that this will affect systemic blood pressure. Because, as you'll see on Wednesday, that we're going to go outside and we're going to start working on other parts of our body that will be affected and not just intrinsically. So what are we going to do when filtrate is high then? So, okay, so if filtrate is moving rapidly or has a high sodium chloride content, then the vasoconstrictor, the macula densa will make a vasoconstrictor for the afferent arteriole. And then this slows GFR so that the filtrate will have a longer time to be processed. Okay. No, the next exam, Erin, if you got to pull up your modified schedule. So the modified schedule is the 24th. So Erin asked, the next exam is showing a week from today. Is that correct? So I'll go back to the, if you haven't downloaded the modified schedule syllabus, it's in the module and it's in Canvas and it's in an announcement and it says the new syllabus or whatever. So yeah, it's the 24th. So we've got a while. Okay. Okay, so that's that. Oh, heavens, no, it can't be next week. Okay, and then sympathetic control. We briefly saw this in the urinary system physiology lab stuff, but always know that sympathetic control is always going to cause vasoconstriction, because remember, sympathetic fibers go to vascular smooth muscle and cause vasoconstriction. So when no sympathetic stimulation is occurring, renal vessels are maximally dilated and the kidney takes care of itself, what its GFR is, through intrinsic control. But during times of stress, so this is the important thing right here, during times of stress, and it can be a variety of stresses, we don't know, could be hypovolemia, but during times of stress, blood is shunted away from the kidneys and filtration is inhibited. So if you think back to when we were looking at the distribution of blood throughout your body with the basketball player guy, and we saw that when you're in sympathetic stimulation, you have enormous blood flow to other parts of your body, and we went to the kidneys and we like decreased it by half. And yeah, it's because we distributed the blood to your skeletal muscles, to your heart, to your lungs, to your brain during that sympathetic stimulation. But then your kidneys are not necessary. They're not a player in sympathetic stimulation at all. So that's that. Oh, whoops, I forgot to clear my drawings. All right, so we're not going to do this, not going to do the renin-angiotensin mechanism right now. What's next? Oops, sorry. Yeah, that's what it wants us to do. So we're not going to do that. So this is a good place to stop. It's 4 minutes. So does anybody have any questions about anything? Like anything? Where am I? Hang on. There we go. Stop sharing. Oops. So here's the new schedule. And so there it is down there on the 24th. Nope. Yeah. Right there on the 24th. Okay. Well, sweet. If that's all, I don't want to go any farther than that. And so if you haven't taken the exam, make sure you get that sucker done today, and good luck. And then so we'll see you tomorrow morning at 8 o'clock. No, tomorrow morning at 9 o'clock and for lab. And we will be doing fluid and electrolyte. Oh, guess what, friends? We're doing acid-base ABGs tomorrow. I'm just trying to think if there's anything that I want you to bring. No, just we're going to do ABGs tomorrow, and it's going to be an exciting thing, which reminds me that I'm going to have to post something. Yeah. Oh, okay. So here's what I want you to bring tomorrow. I knew there was something I wanted you to bring, but you don't have it yet. So in the module, I'm going to post an ABG packet. And I want you to download that packet and bring it to lab tomorrow. You need to have that packet because I need you to follow along as we do things. So I'll post that right now. And so make sure you bring the ABG packet to lab tomorrow. Okay, other than that, I got nothing. So have a great, and yes, we are finishing the other one. So bring both packets. So you'll need this one. I'm gonna go make the video right now. Okay, so we're gonna start in here where it says something, fluid electrolyte acid base balance. So we're gonna work on this packet, rest of that, and then we're gonna start ABGs. So yeah, thanks, Bryce. I didn't think about telling you to bring that one too. Okay, I think we're good. So bye, see you tomorrow.